not Lale. Lale couldn't make it because of visa issues, so I'm going to talk on behalf of her. I am not a biologist, I'm a statistician. So I know that most people here care a lot more about the biology, and how we actually do the technology, the computation to get there is something of a detail that you usually suppress. And I decided that instead of pretending to know what I don't know, it's better to tell you what I do know, which is maybe the boring part. So when the problem was presented to me, we wanted to quantify direct dependence between two residues in the protein. And I was told that this is important for biology because it is deeply related to the allostery process. Allostery is the process which describes a change in conformation in one position. How does that affect changes in another position? So in order to understand this process, we'd like to computationally characterize direct dependence. And our approach follows somebody else's study, Dubai. We focus on side chain fluctuations alone. So side chain fluctuations here, how does that affect side chain fluctuation in a different position? And we like to rank these pairwise dependence. So how do we do it? So here we have two vectors. Let me see. X is a number of dihedral angles describing the conformation of one position. Y is the dihedral angles, maybe a couple of them, describing the conformation of another position. And X could be 4 angles, or Y could be 3 angles, depending on the type of amino acid. Some are bigger. And Z are the dihedral angles of all the other residues, so inside the protein. So that's a big number, lots of dihedral angles elsewhere. So basically, what we're trying to do is we're trying to characterize the relationship between X and Y, but while we're characterizing this relationship, we also take Z into account, so the positions of all the other angles inside the protein. And we do this by using a mathematical concept which we sort of synthesized. It's called Kernelized partial canonical correlation coefficient. So correlation describes X and Y, which is one position and another position. Partial means we take into account everything else. Kernelized means we use a kernel version, and I'll explain why that is. So let me explain the math to you. So everybody knows what correlation is. So you give me two quantities, I can tell you how well they're correlated. So height is correlated with weight, for example. So in order to get to our kernelized partial canonical correlation coefficient, we basically blend in 3 different kind of concepts. The first concept is partial correlation, the second one is canonical correlation, and third, we kernelize this. So the 3 steps, let me explain what that is. So the notion of partial correlation is really quite simple. So if 2 quantities are correlated, but they may not be directly correlated. X could be correlated with a bunch of other variables, Z. Y could also be correlated with a bunch of other variables, Z. And for that reason, you see that X and Y are correlated. So we're more interested in the direct correlation. So the way we do this is we first do a regression. So we explain the changes in X by Z. We also explain the changes in Y by Z. And then we take the residual. So after having removed the effect of Z, now we ask, are X and Y, the remaining part, the residual part, are they still correlated? If they are still correlated, then that says something about their direct. So this is the notion of the partial correlation in statistics. Now the second one is more challenging. I just said if you give me 2 variables, let's say height and weight, I can characterize the correlation. Is it 0.7, 0.8? But here we have a number of dihedral angles at one position, 3 or 4, and another set of dihedral angles at another position, maybe 3 or 4. So now what does it mean to say, how do these 3 or 4 numbers correlate with those 3 or 4 numbers? Right? Now, of course, you can say, oh, well, how does the first number here correlate with the second one? How does this third one correlate with the first one? You can have a matrix of this kind of thing. Of course, that has a lot of information, but it's not very convenient. We want one single numeric number to characterize the correlation. So here we use a notion that is actually fairly old in statistics. It's actually developed in the 1930s by Harold Hotelling. This is called canonical correlation coefficient. It's a very standard multivariate analysis kind of tool. And the idea is the following. Let me now take a combination of these 3 or 4 quantities on the one side, and also allow myself to take another combination of the 3 or 4 quantities in the other side. And I take the combination in such a way so that after I take the combination, I've maximized the correlation on both sides. Okay, so here I have 3 or 4 quantities. Here I have 3 or 4 quantities. I take a combination, so I basically merge this into one quantity. I take another combination, I basically merge that into another quantity, and then I do it in such a way that after the combination, I search for this kind of optimal combination. I've maximized the correlation on the two sides. This is called the canonical correlation coefficient. So this is what this all the ugly math is saying. I choose the u on the one side, that's the combination. I choose the v on the other side, such that afterwards I have maximized. Okay. So to do this, we basically go from the definition of what correlation is. We discover that this is dependent on the covariance matrix between the x and y. These are multivariate measurements. It also depends on the covariance matrix on the x side, covariance matrix on the y side. And of course, we can, if we have enough data, we can estimate the covariance matrices. So we can estimate the covariance matrix between x and y. Here it is. We can estimate the covariance matrices on the x side and on the y side. And once we estimate them, we plug it in, we get a quantity like this. Okay. Now, so this is the idea of the canonical correlation. Now, to merge what I just said earlier, partial correlation with canonical correlation, the idea is very simple. You can apply the canonical correlation not just directly on the original measurements, the x and y, but you can apply it to the partial quantity. So basically, you can take the effect of z out of x, you can take the effect of z out of y, and then apply this kind of canonical correlation idea to x and y, and you've basically merged the two ideas. So this is solved by an eigenvalue problem. So everybody here probably knows about principal component analysis. So here the first line is what the principal component analysis does in mathematics. You have a symmetric matrix. You are trying to find a combination, a certain combination, so that you maximize this quantity, and the constraint is that the combination, the vector, has norm 1. Now you can successively generalize this eigenvalue problem. So here's a slightly more generalized version. You still have the same quantity you're trying to maximize, but here the constraint is instead of a norm being 1, it's a slightly more generalized norm. And then this is the most general form, so you don't have a symmetric matrix anymore, so you're doing something on the left-hand side, you're doing something else differently on the right-hand side. You're trying to maximize this, and the constraint is that on the left-hand side you have a constraint, on the right-hand side you have a constraint. So this is successively more general, but nonetheless, they are all eigenvalue problems. So the problem that we end up solving is actually the third type. We're trying to maximize something on the left-hand side, something on the right-hand side. There's a constraint on the left, there's a constraint on the right. So it's the third type of, so we can do this eigenvalue problem, standard eigenvalue problem. So the third idea is kernelization. So this really was sort of revitalized a few years ago by the support vector machine. A lot of bioinformaticians are using it. So the idea of is using a kernel version. So the idea is if you have a multivariate technique where you see that your technique depends on this type of matrix, inner product matrix called the Grant matrix. So the entries here are the inner product between the first observation and the first observation, the inner product between the first observation and the second one, inner product between the second one and the first one. Of course, it's symmetric. So if you discover that your multivariate technique depends on this type of Grant matrix, then you can replace the inner product by a kernel function, like this. So replace the inner product with a kernel function. The argument is you are essentially still, these entries are still inner products, but they're inner product in a different space. It's the induced feature space, so-called induced feature space by the kernel. So it's a different way of measuring similarity. Now, the reason why this is very important for our particular application is because we are measuring angles. So these angles, let's say one angle is negative 180 degrees. Another angle is positive 180 degrees. Now, on the Euclidean line scale, these look very different, right? Negative 180 over here, positive 180 over there. They're as different as they can be, but in fact, they are the same. So you have to take into this kind of thing into account. So you have to measure similarity differently. You can't just measure similarity using the standard inner product matrix. So it's very natural that you have to replace it with a kernel function. I'll tell you what kind of kernel function we use. So we merge these kind of things together. So here is the original canonical correlation eigenvalue problem. And the way you see that it can be kernelized is that you look at the dual representation. So this is the dual representation is essentially reparameterize the U and the V. By the way, the U and V are your choice. You're trying to maximize. You're choosing U and V to maximize this. So you reparameterize this, and you see that this problem depends on gram matrix on the X side, gram matrix on the Y side, here again, gram matrix. So basically replace, every time you see a gram matrix, replace it with kernel. Now, the only technical detail here is once you reparameterize them in the dual space, you've increased the search space a lot bigger. Originally, these are p-dimensional, q-dimensional, so p could be 3 or 4, like I said, q could be 3 or 4. But once you reparameterize the alpha and the theta, they're in n-dimensional space, so they're much bigger. So you basically increase the complexity or flexibility of your model, and you end up overfitting. So what you have to do is very carefully, you have to put some regularization into it. So this is the technical detail. You have to put some regularization into it. So those are the three pieces of our technology. So we want to compute this. So it doesn't matter whether you understood the technical part or not. If you want to compute correlation, even the basic correlation, let's say height and weight, you need a bunch of observations. I can't just look at your height and weight alone and say, oh, I think height and weight are correlated 70%. You have to look at the height and weights of maybe at least 100 individuals. And then you can see, as the height tends to go up, the weight tends to go up. That's what we mean by positively correlated. So here, we are trying to characterize a type of dependence between two multivariate quantities, but we need to have multiple copies of this. So what that means is we need to observe the protein in many, many different conformations. And then we can characterize, well, what if, what if I change something here? What's going to happen there? So we need a data set. So where do we get this kind of data set? And then the second choice is, well, what kind of kernel function do we choose? So the data set we got in the cheating kind of way. So we grab a protein from the PDB, randomly pick about 20% of the residues, fix that to a rotamer conformation in the rotamer library, and then we use a packing software, such as Squirrel, to pack the rest. Right? And then we do it again. We randomly select 20 positions, 20% positions, fix their conformation to a randomly chosen rotamer position. So there's some randomness, and then we pack the rest. So we do this repeatedly. We get our data set this way. And the kernel function is a von Mises kernel, which has a cosine which takes care of this kind of wrapping around. It's also weighted by, inversely weighted by the energy of the structure, so that more stable structures contribute more information into our procedure. Now, here's something that I want to say about the choice of the kernel parameter. The von Mises kernel is essentially like a Gaussian kernel, like a bell curve for angles. So here's the relationship by using a Taylor approximation in math. You can see this is the Gaussian curve. So in the Gaussian curve, basically what that says is this. If the two angles are exactly the same, then of course you're at the peak. You're very similar. You're the same. And as the difference becomes larger, the similarity drops, right? The Gaussian curve. But there is the standard deviation parameter for the Gaussian curve. So if the standard deviation is larger, then you drop much slower. If the standard deviation is small, you drop much faster. So we can choose these equivalent standard deviation parameters in such a way that the first two dihedral angles, on those dimensions, the standard deviation is about 20 degrees. So they're much sharper Gaussian curves. And later, for the fourth dihedral angle, it's 40 degrees. So because for the outside dihedral angles, you have much more room for movement. So we choose it this way. So our results are, we looked at a bunch of families, small G protein families. This is the part that I don't understand very well. I was told the reason why this is the case is because these are well-studied families. They all have a common structure. They all have this kind of a switch 1 and switch 2 region. And in an allosteric event, a lot of things tend to happen in those regions. So we compare with a contact rearrangement network. This is the work of Daly, 2008. We also used a off-the-shelf sort of method, statistical method that's reasonably new, called the graphical lasso, for characterizing the dependence of two positions. And we actually did this ourselves in an earlier paper. So basically, the result is, well, these AUC, area under the ROC curve, they're about 70%, so 50% is random, 90% is really good agreement, 70% is good agreement, but obviously not as high as 90. But if you have a biomarker for cancer, 70% is considered a good biomarker. So this says the way we do this, we get reasonably good agreement with contact rearrangement networks. And also, the other take-home message here is we are slightly better than our earlier method of basically applying a sort of off-the-shelf, blindly statistical tool called the graphical lasso. And we think the reason is because here we have these kernels and stuff specifically tailored toward this kind of data. And qualitatively, our graphs, this is just an example. We have more examples in the paper. You see that the kind of connections, the top-ranked connections we find mostly tend to concentrate between the switch 1 and switch 2 regions. So that's supposed to be a good idea. And compare with our own graphical lasso network, basically the idea is our new method gives us more connected, highly concentrated graphs than graphical lasso. So here is the blue and the yellow are residues from the switch 2 and switch 1 regions. So they're connected together. Here are some summary statistics to show how the graphs are more concentrated in terms of a fewer number of nodes, but fewer connected components, and the node degree, so that means how many nodes are they connected with, they're higher. So these are all network statistics showing that there are more concentrated networks. And that's supposed to be sort of encouraging idea, because intrinsically there's no reason why we do this kind of statistical calculation. We should agree with contact rearrangement network, right? But the fact that it does agree is sort of, wow, it's a surprising thing. So finally, before I end, I just want to make a comment about how we generated the data set. I did say that this is sort of a cheating way of generating the data set. Now, a much better way is to actually use a molecular dynamics or simulation, because one criticism is, you fix 20% of the residue and you pack the rest. I mean, that structure may not even exist in nature, right, that you're working with. Now, we think you probably don't get to observe that structure ever in nature, but it doesn't mean they don't exist. In reality, what happens is these proteins are bombarded by water molecules around it. And then sometimes they will take on a weird position that you don't really get to crystallize or see, right? Because they're very transient. But the water molecule is bombarding the protein molecule, and then suddenly it takes this position. So what we're doing, randomly fixing 20%, it's kind of a cheap way of simulating, well, suddenly these things change position just because things are hitting it. Well, then we ask ourselves, well, how will the rest of the protein respond? So this is what we're trying to simulate. But we're not really simulating this process, what I just described to you, in the right way. The right way is probably to do molecular dynamics and stuff. And the reason why we didn't do it is just because at this moment, molecular dynamics is so much slower, and the PhD students just can't afford, because that'll take forever to get her PhD. So that's our comment. But it definitely, our method is modular in the sense that you can create your data set more properly this way and still apply all of this and see what you get. And we certainly are hoping that we will get to do that kind of thing in the future. Okay, so thank you.
Thank you very much. We have time for a quick question. Let's thank the speaker again. Thank you.